It's really a lot of fun for me to stand up here and talk some science instead of all the other things that we do. And, I have several projects going on in my group that involve tranSMART, and one of them is developing new methods to extend current methodology in psychiatric pharmacogenomics. And I'll tell you a little bit about that as we go on, and where the activity is with the tranSMART platform, both in the validation phase on a fast follow with our randomized control trial that we're running right now with the GeneSight, and then also with the new work to develop the concept for a new set of single nucleotide variation markers for more precise, pharmacogenomic assay. This talk is dedicated to this guy here, David Mrazek. He invented the field of psychiatric pharmacogenomics and then sadly passed away last year. And I was the chair of the technical advisory board of GeneSight Health at the time, and David was the chair of the scientific advisory board. And it was just really sad to see him go. He had suffering from brain cancer, and just when he had just gotten his home in Hawaii. And he was our leader. I got a chance to meet him when he was still at George Washington before he went to Mayo. And actually, at that point, I was in the psychiatry department, and the chair of psychiatry locked me in a room with this guy for 3 hours and said, "When you come out, you're going to tell me what he's talking about." I must say, at the time, I had a headache, but I finally figured it out. So I just wanted to show you the present and future trends with pharmacogenomics as applied just in general, and then diving down in psychiatry, what's happening now with the tests that's out there with our partner GeneSight, what the future, and this is a collaboration, a deep collaboration that's growing actually quite a bit between the University of Michigan and our department and the company. And then where the platform fits in to the company's plans, both on the validation side for the randomized, following on from our randomized control trial across 12 sites in the National Network of Depression Centers, and then as a discovery platform to enhance our research across actually several research sites, in North America. Here's just some basic. I thought I would just look at some existing and thinking about pharmacogenomics, existing and future trends, and obviously we're into the future, and we would like to see a platform like the tranSMART platform help us reach our future vision. And so we're much more into data discovery, obviously, and that's the sweet spot for the platform. Systems pharmacology, I see Nancy is here from the Reagan-Udall group, and there's some systems pharmacology work that we're getting ready to launch with the FDA. Having more machine learning and learning machines, and ultimately allowing us to stratify, as opposed to just taking in heterogeneous clinical trial participants and not have the ability to stratify the participants. And ultimately developing a knowledge base, not only for the research and development efforts, but those efforts that are involved with the preemptive phenotyping that we're already seeing in some clinics. I think the work at the Vanderbilt University Medical Center, and more broadly the Pharmacogenomics Research Network funded by the NIH are good exemplars, and Mayo has a good program there too. And so one of the things, there's a lot of words here, but in the first generation of pharmacogenomics as applied to not only psychiatry, but cardiovascular disease, blood, metabolism, and so on, is related to exomic analysis of key enzymes and key pathways. And there's a lot more than exomic analysis that's available to us, and I'll show you what we can do with exomic analysis in a combinatoric assay, but the future, the potential for more specificity and frankly more power in the tests. And it has to do with looking at enhancer regions and non-coding RNAs, not just the coding elements, SNPs within the enzymes. And so, and then of course, bringing in other omic data, that it could include transcription regulation, using REST-seq, looking at the transcriptome more, and looking at the metabolome quite a bit. And so you can imagine a day where we're not only taking the cheek swab and measuring, a panel, but we're also measuring blood and urine at the same time. And you'll see why. So, mental illness is, I don't think I have to tell you, but, just in case, it's a big problem on the burden of disease in in our healthcare system. I mean, compared to some of the other cancers, coronary artery disease, the big three down here, we've heard more about PTSD with the folks coming back from the war theater and so on. Depression, the burden of depression and bipolar disease and this kind of thing in society is huge. And this is the area that we focus on with our developments. There was a very important trial some time back called STAR-D, back 2007, 2005, 2008, in that time frame. What STAR-D taught us in psychiatric research is that, the first drug that you take against depression will be the one that has the best possible efficacy, and after that, the efficacy drops out. And so, what you run into is this treatment intolerance as you take more drugs, and this is simply a score of, how kind of a measure of your depression index. But oftentimes one of the challenges that we're facing with pharmacogenomics is that they're coming in. The patients are coming in, they're presenting with treatment-resistant depression, and you'd like to get them on the right medication. The value proposition is there something that you can do to measure, combinatorial assay of genes, both in the pharmacokinetic space and pharmacodynamic space, and make an assignment for what, drug is best to take for people who suffer from depression. And, STAR-D provides the baseline for thinking about that. And but there's a lot of factors actually, and pharmacogenomics is just one of the things that allows for personalized medicine, selection. But, there are a number of other factors that are involved in, adherence. That's one that you can talk about. Just the other day I went in for a routine medical procedure, and, they printed out this output from the electronic health record that we're so proud of, and they said, well, are you still on these medications? And I'm like looking at these things, and, there wasn't something for, heartburn, no, something, are you taking your vitamins? I mean, it's what's in the record and what we're actually doing in terms of taking the drugs and adhering to the drugs are two separate matters. That's just one example. But what we're focusing is on here now. And, so just for those of you who are not into this game, there are two kinds of genes that we look at here. Genes that are involved in modulating things that are happening at the level of receptors in the brain, like the SSRIs and things like that, serotonin reuptake inhibitors. And then looking at the so-called pharmacokinetic genes that are, cytochrome P450 series that are in your liver. And what we've done here is we've looked at, here's some PD genes that are related to the metabolism of certain psychoactive medications here, okay? Here's some, here's an example of the serotonin transporter. Here's looking at high activity, an ultra metabolizer, a low metabolizer, a medium metabolizer, having to do with, various genetic, things that you can measure. It could be SNPs, could be copy number variants, could be things like that, and we can measure those things. This is just one example. Looking at, so the phenotype is, how do I respond to this drug? The genotype is a series of different measurements that you make from different substratified cohorts in the population. And here's a list of the different neuropsychiatric drugs and what enzymes within the liver are contributing to the metabolic profile of that drug in a human being. Okay, adult human beings. And there are gender differences, and there are age differences, and there are differences having to do with how many drugs you take, the so-called polypharmacy effect. All those things that need to be taken into account. And here's some cytochrome P450 genes. This is a long story here. We won't get into the details. And some of these names you might or may not recognize, but you would certainly recognize them if I called them Zoloft and Abilify and some of those other terms that, you know, they're actually code named. You know, and so we can actually look and we can classify the various genotypes from the ultra rapid to the poor metabolizer. Yesterday at the restaurant, our colleague Michael Braxenthaler ordered a double espresso at 11 at night. Of course, he travels around a lot, so we don't know what time it was. But, you know, we said, gee, you're going to have a double espresso at 11 at night? You know, sure he is. You know, he's probably an ultra metabolizer of cocaine, or of caffeine. Now that too. You know, and the next person at the table won't touch the stuff, won't even look at decaf. And so we do have these different capacities. It's just a simple example. But we also have these capacities for how we metabolize psychiatric drugs as well, both having to do with the liver and having to do with the PD genes that we can measure in the brain. You know, one of the things you've got to do is you've got to also, you know, do some detailed modeling in the metabolomic pathways that are involved with this. And oftentimes these are quite, you know, detailed, and there's little sub-pathways and this is like kind of an art unto itself. And Nancy, this is something I'd like to talk to you a little bit about. It's very interesting. Won't get into it now. So here's the big picture. You have, you know, all these, here's your, you know, genes down here, and these numbers are increasing. You have 38 medications in the panel. You have some, you know, machine learning and other kinds of algorithms. You ultimately classify patients in terms of which drugs do they metabolize, you know, in an ultra rapid or fast way, which can't they metabolize, and then output that into a chart. So so the healthcare physician orders the test and then classifies it into this chart. This is just basically what, you know, this business is going now. We've got 100,000 genotypes. What I want to talk to you is about the future in tranSMART. So, you know, we did several clinical studies, and, you know, against treatment as usual versus use of the test, we did pretty well on 4 studies. We decided we needed a randomized control trial because we have actually gone to the CMS and got pricing so that we can give this test all across the country now, and also the Department of Defense, the Veterans Administration, and the so-called Bureau of Indian Affairs have also approved this. But we preemptively started a randomized control trial that we are running out of the University of Michigan Depression Center with all these other sites. You know, and this trial is actually underway now. We've accrued about over 220 patients. The accrual will be done before the end of the year. And, you know, this is the first area, you know, I'll show you the second area, but this is the first area where we're putting tranSMART in to follow on, because now that we've built this network, we can use this recruitment network to validate new findings that we're coming up with in the lab, which is also going to use tranSMART. So let me take you to the new work, okay, and what we're up to. Okay, and this is work that I've been doing closely with Gerry Higgins, who is a VP of Pharmacogenomics in the company. He's actually half-time joining our faculty in our department now. He's moving to Ann Arbor. He's a long-time collaborator. Okay, what we're into now, and you guys, a lot of you might recognize this, this is looking at the chromatin structure that's underneath the genome. And, you know, so in the earlier days, Dr. Mrazek focused in on these exons in the various genes for the PK and PD. You know, everything in our test was based on the exons. But it turns out, you know, that the regulation of the expression of these genes, both in cis and trans, has to do with what's going on up here in the so-called enhancer in the transcription regions. This is called the regulome. You know, so there's things that are regulating the expression of these genes. You know, and the question is, what's happening there? So we started to take a look at that. And so we use the NHGRI GWAS catalog. We actually use 2 sources of data. The NHGRI GWAS catalog, and we took down all the data that came from pharmacogenomic studies, you know, that are actually from, you know, and we took down 14 studies and just looked at the SNPs within those studies that were published on the NHGRI catalog. We also drew down 12 psychiatric pharmacogenomic studies. So we take all those variants from all those studies and all that data that's coming along for the ride, and we started to put those through a pipeline that would allow us to look at the SNPs in the so-called enhancer region. So we actually, there's a guy in our department now that we just hired, Alan Boyle, who's one of the pioneers in this area of gene regulation in the Regulome. There's another guy here, Dick McCombie, who, it was a graduate of the University of Michigan way back when. We reconnected with him. He's at the Cold Spring Harbor Lab and runs the Stanley Institute for Psychiatric Genomics there. Edwards et al. We started to learn how we could take this data and reclassify the SNPs and understand, where those SNPs were actually mapping to the genome. Were they mapping in the intragenic region? Were they mapping in the intron? Or were they mapping in the exons? Everything that we were doing with our test and are doing today is in the exons. Well, here's the interesting story. This is actually data that we pulled from a review article by Zanger et al., who was expert on the cytochrome P450s, and we recalculated their data, and we found that most of the single nucleotide variants are in the non-coding regions. So, that's the exons there in the coding regions, but most of the actual variants that you can measure are in the non-coding regions. And it turns out this is the case in all diseases and conditions. This isn't just psychiatry. This is just what we do in our research. This is general. This is diabetes. This is everywhere. So we've been looking at the exomes, like in cancer. We did a lot of work early on with the rule and the gene fusion events. We're all looking at exomes. I'm talking about the introns. So in the liver, we already found from this group Zanger, this was the story. When Gerry and I took the data from the NHGRI GWAS catalogs, we found the same kind of thing. The intronic region and the intragenic region, had about, 80% or so of the single nucleotide variants. The rest of the variants are in the coding regions or maybe in the promoters. And this is the rule of thumb that everybody seems to be finding now, that, most of the SNPs that are associated with disease are associated in the intragenic or intronic regions. Now, you can go actually a little, and so there's the Psychiatric Association studies there. Now, you could go further, and you could actually map where those SNPs are, in these intragenic. This is just looking at intragenic regions. I don't know, Gil took off, but he's really good on the splice acceptor site stuff, which is really more the intronic stuff. We're seeing a lot of neat data on bipolar disorder, and that, it'd be alternative splicing. That would be the mechanism there. But what's happening, in the intragenic regions is modulation of the gene regulation, okay? That's what's happening there. And so what you can do is then you can pull that data down further. I'm not going to show you that right now, and relate it to cell line data from ENCODE and tissue data from the Allen Brain Atlas and from the Broad Institute. And you can start to actually see associations between these SNPs and, modulated drug response in certain tissues. That's for another day. But what I wanted to show you, because I wanted to try to get back on schedule here, is what the UMich and AssureX are doing in collaboration. This is just something that's starting up now. And we've got a guy coming back to Michigan from New York City, Ed Barber, who's currently the director of informatics at the Rockefeller University, will be moving back to Ann Arbor to work in the lab and work with the company so that we can build this. And Nancy, again, this is something I think I really would like to see. I think there should be some interest here. What we're building is a tranSMART instance here that will, and I'll show you the version 1.2 applications that we're starting to get up and running right now to test against this. But what we want to do is develop a instance of tranSMART that would allow us to take, these are some of the, you can see already Cytoscape is something that is interoperating through the API of tranSMART. We've been using that for years. We want to continue to use that. You can see the Pearl and the bioconductor is already in there. GeneVar, ClinVar, that kind of stuff. These are what we need. HaploRag, Metacore, that ought to ring some bells with people in the audience. So these are some of the analytics and knowledge bases from companies we want to put in. The old PharmGKB, which is now maintained by Personalis. So what we're doing is we're making a specialized tranSMART instance to enhance the research that we're doing with the variants that we can see. So you can see down here the Pfizer GWAS, PGEX. So one of the first things that we're just gearing up to do now, Mateo, is we're going to run this analysis again with the Pfizer tools and see how it stands up to the GWAS tools that we've been using, as an example. And this goes back to Garrett's point. We've got one with the Hopkins project now, too. It's that that pie chart that Garrett showed from the Collins and Tabak paper on the reproducibility of data, that's actually in the wheelhouse of this group because this is clinical research that they're talking about. And the reproducibility of the data is not, of the studies, has been problematic. And one of the things that we feel is a really source of low-lying fruit for us in the science right now is to reproduce old papers that we published with one set of tools and methods with the new set of tools and methods and compare. And Gil has been after us for years to do that. And we've been publishing a lot of paper. Like, we developed a interactions database, and set of tools years ago. Well, how does it compare to David? How does it compare to other tools out there? How does what we're doing compare to what we did before in doing, kind of side-by-side studies to, bootstrap up there and see the level of reproducibility. So this is, and we intend to make this open to the community. We already saw one example up in Montreal, and we were very excited about that because there's a very big effort right now with the company, with collaborating with Jim Kennedy up at the Center for Addiction and Mental Health Research in Toronto. There's a 20,000 patient genetic study that we're doing in Toronto that we're also going to run through this. So this will be available for the discovery side of the shop. And we will bootstrap this up. We'll have our own website and make this available to everybody in the community. So anybody in the neuroscience, I don't see Magali here, anybody in the neuroscience area will be able to participate in this more as a team effort. And then, you take down your own instance and then go from there. So that's the plan. And, there's a big contract deal that we're putting together right now. In fact, it's being, this work with the SureX, as I hear, going to be approved by the regents in 2 days. So we're really making progress. In case was very helpful, obviously, in getting this whole thing going. But, I really got more interested in using this when we realized that, we could wrap the different formats because we're really looking at epigenomics. So the question is, why would we want to use this browser? Why don't we stay in Santa Cruz? But we can do everything that we want to with this. We can make this look like a customized Santa Cruz browser. And that's what we're going to do. And any changes that we make, we'll throw back to the community in the spirit of, our sharing. But this genome browser we feel is going to be very helpful to us. In all the analysis that I showed you, we used the Santa Cruz browser. So this will be a one case-by-case comparison. We've got several manuscripts that are either gone out for review or being prepared and sending out right now, and we want to put this in the method session. Now, that dirty little three-letter word that Garrett was mentioning, there are specialized ETL pipelines, we assume, that will be needed for, pulling data from Pharm GKB and other sources. And, this is still something that Ed DeBarber is going to be working on this one. But, we would like to use what's out there and then contribute more into these ETL pipelines. And this will be something we'll talk about tomorrow in the content committee. It's obvious why we want to use, first of all, the Pfizer. When I attend these pharmacogenomics workshops in the West Coast, I'm always impressed by the quality of the GWAS work that's coming out of the Pfizer labs. It's absolutely first rate. And so we're very excited to be able to use these tools now, and we will be implementing these straight away. And, doing some, we've got lots of expertise here in GWAS, here locally in the community with Consuelo Abicasis and Mike Banke and Julie Douglas and several experts. So, we'll give it a good run and do a good evaluation of this. I'm very hopeful. Sample handling with our national network for the Depression Center's partners, we'll put in a lightweight scheme that will allow us to handle not only the cheek swabs, but the bloods, and we're interested in monocytes, and in whole blood and urine. So it'll be a stripped down version. And so, you know, okay, good. I wanted to cut it down a little bit to give us time to get back on schedule. And it's early days for this, but there's a commitment. Jim Burns, who's executive chair of AssureX, was here and participated in the board. There's a commitment that the company has in our partnership with Michigan and beyond and the tranSMART Foundation. You know, what we have been running into is this silo mentality in this field. And, you know, one of the things that's driving us, of course, is the Food and Drug Administration. So if you look down the road and see in terms of, and Ike had gotten a nice discussion going in personalized medicine at the board meeting. You know, we are going to have to share. It's not going to be enough. Here's your report. You know, you're green, yellow, red. You know, we're going to have to share with the patients, you know, what findings, what single nucleotide variants did they find? I mean, this is coming. Having the ability to share data with the patients and with the providers at scale. And we feel that, so, okay, you might win this one. I mean, you know, we're doing this for research now, but one can see the day that a fully validated form of this and flipping a few switches could even allow us to share this data more broadly as we develop it. And so that's our vision for working on the future of psychiatric pharmacogenomics and how important the tranSMART platform is to our vision. You know, if our leader, Jim Burns, were here, and Gil has heard him say it many times, this is a bioinformatics-based enterprise now. It's really changing, and to have the bioinformatics platform to do this kind of research and translate it into practice rapidly is a commitment that we have, and we feel very strongly that it's going to do well. I hope this has been helpful. Thanks. Yeah, oh yeah, yes, hi. Yeah, I can hear you. Oh, where's that microphone? Here he comes. He gets some good exercise with it.
Can you comment on what do you plan to do with those SNPs that you identify? What is your plan for downstream analysis? And specifically, if there is any way you envision how you can compare gene transcriptomic data with this data?
Well, this is great. This is a very, you know, you put your finger on it, because ultimately gene regulation is one level, but transcription regulation is the thing that's going to, you know, take the transcriptionally, you know, kind of inactive chromatin. You have the transcriptionally active chromatin. And what we're really talking about is transcriptionally poised chromatin. And, you know, the key to success, and I didn't get this in here now, but we've got a lot of preliminary data, and we're just trying to get this Regulome stuff out the door in a couple cases, is transcription regulation. You know, and we're actually working on that. You know, one of the issues that we have is that in order to actually do that, we've had to go into proprietary databases like Transcript. It's really TransFact Pro that we're relying on there more than anything else to give us good data on transcription factors that might be, you know, key in flipping that over into transcriptionally active.
Actually,
yeah.
I think I have BG right here.
So that's a great question. You're really, that's the next step, and we're working on it.